Thanks for coming. My name is Fer. I'm the EMEA FAS manager. And today I'm here with an impossible mission. I need to give you an overview of who Twist is and our portfolio in 15 minutes. That's not going to happen. I don't want to overwhelm you with data. I just hope I'll give you an insight into who we are and what we do, and into some products that might be relevant for the audience here today. And I raise some interest on you so that you will come by our booth and ask more specific questions related to your product, to your project, and how we can collaborate together. Twist was born 10 years ago in San Francisco with the mission of providing our customers tools to help them to improve healthcare and to work towards a more sustainable future. What we do is synthesize DNA, but contrary to our competitors who are using 96 well plates, we use a silicon platform that really enables us to scale up the process. So you might ask yourself, what are you doing in an NGS Congress, which is about reading DNA? Well, around 5 years ago, we realized that we could use our technology to design and synthesize oligoprop panels to use in enrichment workflows. We first began with this, but then we developed workflows, both library prep and target enrichment workflows, that will enhance our oligo panels to get the best out of them. Nowadays, we have fixed panels, and then we also work with leading institutions to develop alliance panels, but we reckon that sometimes this does not meet the needs of our customers. So in those cases, we develop together with you custom panels more suited to the specific targets that you are looking for and to the specific goals of your experiments. This has enabled us to help in many different applications, like, for instance, basic research, diagnostics, and more recently, epigenomics and transcriptomics. Why would you want to use target enrichment? Well, the main reason is that it's going to help you adjust your budget. It's going to save costs on sequencing. As an example, if you are interested on the human exome and are to do an enrichment experiment targeting 20x coverage, with enrichment, you would need 7.5 gigabytes of data. If you were to do whole genome sequencing, that will mean 90 gigabytes of data. As you can see, the reduction on data is drastic. And what that means is that with all that data that you are saving, you can just add more samples. This is going to be key, as I said, for cost savings, but also for some applications where you're looking for very low frequency variants, like, for instance, if you're in the cancer field. It's really going to be a limiting factor to have a lot of reads, and enrichment will allow you to get this. Some scientists will say, you know what, I don't trust on target enrichment because you just introduce bias from props. And this is what Twist has been working from the very beginning. We have developed a proprietary method to synthesize and amplify props in a very uniform manner, so that when our competitors are synthesizing props with many biases towards GC content, our prop synthesis is going to guarantee you that you have the same representation of all of the props within the panel. This has a direct effect on the end results that you're going to be getting from the sequencer and on your data, in the way that all of your targets are going to be equally represented from the sequencing reads. This means that it won't happen to you to analyze the final results and look for one of your targets of interest and find out that in some of your samples, you just don't have enough coverage and you need to resequence the sample. That is very annoying, that increases cost and adds times. So today, as I said, I just want to go through a few of our workflows that might be interesting to you. The first one of them is sample ID. I don't know if it sounds familiar, the situation of processing many samples at the same time, patient samples, and then at the very end of the workflow, when you get the FASTQ files, you realize that there was a mistake along the workflow, and the data that you have does not correspond to the patient that it should be. This is a catastrophe. And that's the purpose of this kit. So basically, it is a very fast workflow that will enable you from 2 microliters of the starting blood sample from the patient. It will amplify 30 significant SNPs, and then you will adding to the sequencing run a minimal proportion of this sample ID prep. What you want to have at the end is those 30 significant SNPs concordant between your sample ID sample and your proper patient sample. If that's the case, you're good to go, and you have the peace of mind of knowing that it really is what it should be. Another application that we have been working on is on long reads. Long reads some years ago was not that known. Very few users were actually benefiting from it, and it was not so well known. But as years pass, the relevance of this technology is increasing. As you can see, it was named the Method of the Year in 2022. And the reason for this is because it is really enabling customers to fill up the gaps that the short reads have. Those difficult to sequence regions that with short reads you won't be able to align just because of the nature of the reads. There are, like for instance, repetitive regions that the aligner won't be able to actually discriminate from one or another. Long reads will enable you to do these alignments. This is going to be key, for instance, for some structural variants as well. And for haplotype calling is a very handy method to follow. We have worked together with PacBio to develop a workflow that will help you to do long-read sequencing on enriched targets. Again, the goal is going to be able to focus the reads that you're sequencing only to your targets of interest. Initially, our customers were using custom panels for this application. So basically, they were partnering with us to develop the panels based on their needs. This year, we launched 2 alliance panels, the Dark Genes panel, which is going to be focusing on difficult to sequence regions, and the PGX panel, which is focusing on pharmacogenetics targets. What you can see in this slide are just few examples of different size panels and the number of samples that you can sequence on a couple of different PacBio sequencers, so that it will give you an idea of what is the resolution that you're going to be getting. One note to make here is that this protocol, as I said, it was co-developed with PacBio. The truth is we have more and more customers using this protocol with ONT successfully. So this gives more flexibility to the customers. Our methylation detection system. I don't want to get too deep into this because we have 2 speakers today with us that are going to be showing you their results when using this system. Just a brief introduction on it. We partner with NEB to develop this system. Basically, it consists on the NEB conversion and library preparation, followed by our enrichment. This conversion is enzymatic conversion. I don't know how much you know about methylation, but it is a much milder conversion than bisulfite, meaning that you will be able to start with lower input amounts. Adding to this, it doesn't have the bias that bisulfite conversion has towards GC content. Once you have your library preps ready, you will proceed with enrichment. We have developed a special type of probe panels that will maximize the capture of your methylated targets. These are probe panels that have 4 probes per target, 2 against the plus strand, 2 against the minus strand, 1 against the fully methylated and another one against the fully unmethylated version of your target. What this does is really making sure that no matter what the status of your target it is going to capture it. The fact that the enrichment goes after the conversion, it enables you to really go lower on the DNA input into the system, which is a key for some applications where the DNA input is a limitation, like it is, for instance, if you're working with CF DNA samples. Finally, I didn't want to miss on our latest baby. This was launched a couple of weeks ago. It is our RNA sequencing workflow. It is a very fast and easy RNA library preparation kit. Right now, the fastest one in the market. If you decide to do whole transcriptonic, you have an RNA and globin depletion step at the very beginning. But we didn't want to miss on developing targets specifically for RNA as well. So we have came up with an exome, our algorithm, that basically avoids generating probes in boundary between exons. And what this does is that it's going to really maximize the on-target into RNA sequences and not DNA, and it's going to minimize bias towards certain isoforms or fusions. It is very flexible. You can start from 1 nanogram and go up to 1,000 nanograms. You can use all type of quality RNA, from FFP samples to good quality material. We also launch an RNA exome panel. You could also develop your own custom RNA panel with our RNA exome-aware strategy. And we developed, we launched in collaboration with CIGAT, our Alliance Fusion panel, which focuses on relevant fusions for cancer. Since it is such a new workflow, I wanted to give you a little bit more data on it. So over here, you can see the levels of depletion that you managed to get with our rRNA and globin depletion steps. As you can see, it is below 2% the number of these molecules left in the prep after you go through these steps. It is very handy that it is a stranded solution, meaning that you can really reckon which strand from the DNA the RNA is coming from. And then I wanted to give you some insights into the enrichment as well. Sometimes you have to use whole transcriptome, and that's perfectly fine. But if you have the opportunity to perform enrichment, you can see here the number of genes with 5 or more reads, with a different number of million reads per sample. And you would see that at 6 million reads with enriched targets, in enriched libraries, you reach the same level of coverage than if you were doing a transcriptome experiment with 10 million reads. So the cost savings here is, again, quite obvious. Finally, just wanted to show you a comparison of the representation of CDS when you are following an RNA exome enrichment experiment versus whole transcriptome or 3' counting. You can see that you really manage to enrich on CDS regions. And this is all I have to show you today. I hope I raised some interest. And again, we welcome you to have a more in-depth discussion in our booth. If you have any questions here, happy to take them now. Okay. In that case, I'll introduce you to our 2 speakers today. First of all, Julia Henschel from the Human Genetics Institute in Leipzig. She unfortunately couldn't make it today, but she has recorded her talk, and she will be available for the Q&A session after the talk. She's going to be talking to you about the use of our methylation system for the detection of global epigenetic changes in humans and the impact that that has in diagnosis. Our second speaker is Ola from the University of Edinburgh. Again, she's been using our methylation detection system, and she has done a comparison with the EPIC array. She studies the methylation pattern on the effect of smoking on the methylation pattern on humans as well. No further ado, I'll leave the stage to Julia.
So, hello everybody, welcome to my talk about DNA methylation. These are my disclosures. So when we are looking at the use of a single exome analysis in NDD cohort, you can see that the diagnostic yield is about 30 to 30%, depending on the design of the studies. And this is also what we found in our own cohort in the last year, where we can solve 31% of the NDD cases. That means that more than 60% of cases remain unsolved. And if you add genome analysis to that diagnostic pipeline, you can find in literature data that additional 8% of cases can be solved. So what is the reason for missing the diagnosis? Maybe the variant is not present in the analyzed tissue, or maybe the variant is at that low alternative allele frequency that we are not able to detect, so some kind of mosaicism. So maybe the variant could be a repeat variant or structural variant, which is maybe hard to detect in short read sequencing, or it could be an intronic one and the interpretation can be complicated. It could be a uniparental disomy, which is hard to detect in single exomes. And furthermore, it could be a regulatory change, which would also not be able to detect in exome analysis. And we focus today on the regulatory changes. So when we talk about gene regulation or gene expression, there are three major mechanisms, to keep it simple for today, and we will focus here on DNA methylation in my talk. So it means that a methyl group is added to cytosine by DNA methyltransferases, and this often occurs in the CBG islands of promoters, and this leads to gene silencing. When we get, normally we get one copy of each gene from the mother and the father when we only focus on the autosomes, and each of that copies is can be read out when we look for non-imprinted genes. But for imprinted genes, we have copies derived from one patient or one parent, which is silenced by methylation, so it's off. And when we then have a variant on the active copy, that can lead to the classic imprinting disorders. Furthermore, there are genes of the so-called epigenetic machinery. To date, we know about 70 of that genes, leading to over 80 human conditions, and patients affected with this show mainly growth defects and intellectual disability. So these conditions are mainly autosomal dominant inherited, and the variants which you found are de novo loss of function ones in the most cases. So as you can see here, that epigenetic machinery genes can have 4 different enzymatic functions. So are the writers, which introduce methyl groups in the DNA, the erasers, which delete methylation, and then we can have the readers and the remodelers. And there are some of that proteins which can have dual function. And one can imagine that if one of that dual function proteins is affected by a pathogenic variant, then the phenotype can be more severe. So how to measure DNA methylation? One possibility is to do it targeted. So in that case, we use an methylation sensitive digestion and then followed by an MLPA approach. And this is a nice technique if you have a really strong suspicion for one disease, in that case, a Prader-Willi syndrome, and here we can confirm with that MLPA analysis and hypermethylation of the imprinting region of chromosome 15q, which can be caused by a paternal deletion or a uniparental disomy or by an imprinting defect itself. The other possibility is to measure the DNA methylation in a global setting where you don't have you don't need a hypothesis. The DNA is converted, and then you can read out that conversion with microarrays or NGS techniques. So we decided to use NGS because we want to have the possibility to get a single base resolution in the future. And we finished with the ERIC technique some years ago, and it's more easy to have only one technique in the lab, and also NGS setups can be more easily automated on robots. And in the main studies you found in literature, or in the majority of studies, the conversion is done by bisulfite, but we decided to use the enzymatic conversion by twist, because there are many papers already out that show that that an enzymatic conversion leads to a much lower DNA degradation, and so you can get more total reads and more CPG regions covered, and you have better alignment and a higher library complexity. So when we came back to this, the slide from the beginning, then we want to tackle with that technique our unsolved cases. So this is not that easy because normally the settings are that you compare one group against another group and not a single individual against a control cohort. So we have to design a new setting for that. And at first, we have to find the best wet lab conditions. And this work is mainly done by Zenta and Maike from my lab. And we found out that it's nice to have or good to have a high DNA content or DNA input and good DNA quality. Of course, use mechanical fragmentation because the enzymatic fragmentation is not compatible with the enzymatic conversion. And then we enriched this DNA fragments with a custom methylation panel from Twist, which had the same target regions which are on the epic array. Yes. And then it's also important to know that, you have to find out the best loading concentration on the Methyl-seq to avoid duplicates. When you analyze that a little bit further, then you can see there's a cutoff of 60-fold coverage that you need to get good quality data. So that means that you need about 120 million reads per sample to get 90% CPG regions covered. Secondly, you have to find the best control cohort. And for this, we analyzed different tissues because it's known that methylation is tissue specific. And you can see here in the cluster plot that the blood samples have a complete different methylation profile compared to the skin biopsy ones. So it's important that you compare DNA which is derived from the same tissue or material. And the good news is age of the probands and sex of the probands seems to be not relevant for the methylation in our cohort. And here in that cluster plot, you can also see that we had some replicates and they nicely cluster together. So their reproducibility was really good. And then you have to set up a pipeline because actually there's no commercial pipeline or pipelines that you can easily use. So this work is done by Laura and Stefan. And as I'm not really familiar with bioinformatics, I strongly recommend to ask questions directly to Stefan, and I think he will help you as fast as possible. And just to have a small look on the work they did, they set up a pipeline, so-called Nextflow pipeline, which end up with the processed data. And in the second step, they had the analyze Nextflow pipeline, where they identify the potential differentially methylated regions. And they also did some nice visualization where you can compare or where you can set up the control cohorts and the patient, and then you can see that plot where you can here see the a patient in that example and the corresponding controls. So now we want to use that new technique in the lab to add on our cohort. And at first, we start with known imprinting disorders, as I showed you in one of the first slides. So here we have an imprinting region on chromosome 15, and we had some known cases with changes here. So this is a classical Angelman syndrome with a class 2 deletion, leads to an of the maternal copy, which leads to an hypomethylation, which we can nicely reproduce with the EM-seq technique. And we have also some duplications of paternal and maternal alleles, which also leading to hypo or hypermethylation, which we can confirm with that method. So proof of principle, check. In the second case, we used this new technique. We had a exome or panel analysis of a patient where we detect a homozygous causative variant. And initially, we thought that each parent gave an heterozygous variant to the child. But when we reanalyzed our cohort using our new UPD detection tool, then we identified a uniparental isodisomy of the paternal chromosome 18. And this looks like this. We can see a homozygous stretch in that chromosome. And if you want to have more information about the new tool, which is called AlterF plotter, then I recommend to visit the poster of Johanna, which is on the conference. And then we performed EM-seq because there are known imprinting regions on chromosome 18. And we can confirm that there is a hypomethylation in the region of chromosome 18. So this technique can be nicely used in the case of suspicion of imprinting disorders caused by UPDs. And on the third use case, we had a cohort with KDM2A disruption by missense and truncating variants. And KDM2A is a methyl or a demethylase. And when we use the Methyl-seq with DNA from blood compared to the KDM2A group, then you can see that KDM2A lead to a complete change of the methylation profile. And we can now use that technique to analyze variants of unknown significance when found in that gene or in other genes of the epigenetic machinery, and can this use as a functional test. And our next goal is to use that technique on a more or on a higher resolution level to check what is the real effect on the single differential methylated regions in the patients. And if you want to know about this, I can recommend the talk I gave on Saturday, which is now available on demand. So this, to sum up, we can use the EM-seq technique in clinical settings, and now we are ready to use that that pipeline on our unsolved cases. It was more or less easy to implement, and we now can automate it on our robots. It is important to know that you have to set up the bioinformatics pipeline by yourself, depending on your questions. And it's important to have sufficient coverage in your NGS data to have a good resolution and detection rate, and choose the right controls, especially compare the right tissues with each other. And with this, I like to thank to my great team in Leipzig. And if you have any questions, then you can also write an email to me. With this, I'd like to thank you for your attention.
Right now, so if you have any question, please do use the microphone. In that case, thanks, Julia, very much for your talk. It was really insightful. We will move forward with Ola.
Hello everyone. My name is Aleksandra Chybowska. I am a second-year precision medicine student from Marioni Lab, University of Edinburgh. And today I'm going to tell you a little bit about my project in which I use Twist's human methylation panel in order to study smoking and brain health. So as we know, smoking is an important risk factor. It is a well-known fact that it increases the risk of developing more than 50 different conditions. And for that reason, the number of cigarettes smoked per day has been incorporated into various disease risk prediction tools, such as the one shown in this slide. It is a screenshot from ASSIGN, so a cardiovascular disease risk tool developed for Scotland. However, smoking is tricky to measure. Often used self-report data are prone to bias and also don't account for passive smoking. We can use the concentration of serum cotinine in order to measure smoking, but it only reflects short-term exposure. So it may be possible to use DNA methylation information to build a stable biomarker of smoking. A couple of years ago, my colleague Daniel investigated signatures of starting and stopping smoking. And what did he see was that not only that smoking leaves a DNA methylation footprint that can help us to distinguish smokers from never smokers, he also observed that the number of CpG modifications associated with smoking start to revert 5 years since smoking cessation. So in this project, I aim at building a stable and long-term biomarker of smoking. In my analysis, I used data from 2 Scottish cohorts, namely Generation Scotland and Lothian Birth cohort. In Generation Scotland, we had access to methylation data measured in blood samples, whereas in Lothian Birth cohort, we could also use data from from the brain. So the first experiment that I ran was epigenome-wide association studies of smoking, and here the sample size was 80K individuals. Next, I extended this analysis by using the twist kit to look at sequencing data of 24 pairs of smokers and non-smokers from Generation Scotland, and I will soon focus on that part of the experimental setup. And when that was done, I developed an epigenetic score for smoking. I tested it in Lothian Birth cohort, related it to brain health outcomes, and also I compared methylation signatures from brain and from the blood to see if my findings translate across tissues. So first I will give you a broad overview of non-twist analysis, and then I will focus on things that I did using twist human methylation panel. So as I told you before, the first experiment that I ran was an epigenome-wide association studies of smoking. And using Bayesian approach, we identified 26 loci that were significantly associated with smoking. 2 of them haven't been reported before. So the first CpG site that we discovered was annotated to gene GRAK5. And that gene encodes a receptor for glutamate, which is an important neurotransmitter in our central nervous system. And we know that glutamate also plays a role in developing addiction. The second CpG was annotated to a gene called HOXA4, and that gene normally is expressed during embryonic development. However, it can also be abnormally expressed in cancer, and here specifically in lung cancer. So having done that, I was ready to build the epigenetic predictor of smoking, and compared to the previous version of the predictor that my lab published before, we had a better ability to distinguish former smokers from never smokers. And I need to mention here that the epigenetic effects of smoking on the epigenome are so strong that this predictor can almost perfectly distinguish current smokers from never smokers. And it's not only my predictor, the previous versions of the same predictor did the same thing. So this is how bad smoking is. Okay, so in the next experiment, I wanted to relate my epigenetic predictor to the changes in the brain. So we know that methylation is tissue specific, and I built my predictor based on data from the blood. So I was interested to see if we can use this kind of data to predict changes in the brain tissue. And it turned out that yes, indeed, we can do that. We observed that the predictor was strongly associated with different metrics of brain health, and specifically with cortical thinning. And in terms of tissue specificity, several loci showed near perfect discrimination between different categories of smokers, but these didn't overlap across tissues. And taken together, these findings shed light on the mechanisms by which smoking influences brain health. So all these experiments were based on RI data, and we know that RIs measure only a subset of the epigenome. So for instance, the EPIC array currently measures roughly less than 5% of the epigenome. So to get a higher resolution of epigenetic data, we can profile the entire epigenome, but this is a costly experiment, certainly more expensive than using a microarray. So there are approaches that lie somewhere in between using arrays and whole methylome profiling. And one of such approaches is targeted sequencing, and this is exemplified by Twist human methylation panel. So by using targeted sequencing, we can measure methylation at a higher number of CpG sites than available on methylation arrays. And in the same time, the cost of such an experiment will be lower than whole genome profiling. So the Venn diagram showed in this slide shows the overlap between EPIC array and Twist methylome panel in terms of the numbers of CpGs that we measure. And we can see from it that Twist human methylome panel measures methylation at approximately 3 million more sites than the EPIC array. So in my experiment, I used Twist methylation panel to find differentially methylated regions between 24 pairs of smokers and non-smokers from Generation Scotland. For each CpG measured using Twist, we ran linear regression model, which was adjusted for age, sex, and smoking status. Then we executed ran really similar experiment just using EPIC data, and then we compared and contrasted results obtained by the two techniques. So let's start from methylation calling with Twist. I decided to use an R package, an Xflow package called Methyl-seq in order to call methylation in Twist data. Methyl-seq was developed by NF Core community. It was also curated by the same community. And its main functions include FastQ file preprocessing, read aligning, and quality control of the results. As I said, it was developed using Nextflow, which is a domain-specific language for developing scientific pipelines. It can run on different computing platforms, be it your own local machine, high computing cluster, or cloud services. And it also supports parallelization, which is important. It works well with different containerization systems, so you don't need to worry about installing any applications on the hosting machine. All dependencies of the pipeline will be downloaded automatically and executed from within the container. So if you want to find out more about this, please come and talk to me after this presentation. I am also presenting a poster, so we can talk there. When Methyl-seq finishes running, it produces a set of bed graph files. So one bed graph file is produced per sample. And in order to be able to continue analyzing your methylation data, these bed graph files need to be merged together and post-process. And I decided to use an R package called Metrix in order to do that. And my rationale was mainly that there is a best practices pipeline that I could follow to do that. And another advantage of Metrix is that it produces output files in G-ranges format, which can be then easily annotated with another R package called annotator. Now, it is possible to filter your data as part of post-processing of bed graphs. And Metrix has implemented two main filters. The first filter is a SNP filter, which allows you to remove all CpG sites that overlap with C to T polymorphisms. And the other filter is a coverage filter, which allows you to remove all sites covered by a low number of reads. And we want to have high depth of coverage because this increases our confidence in the results. And in order to produce results that I would be confident in, and also keep as many or retain as many CpG sites in the dataset as possible, I experimented with different settings of the coverage filter. So the results of my analysis are shown in the table, shown in the slide, and fields highlighted red correspond to the combinations of settings that result in a number of CpG that would be lower than these available on the EPIC array. The field highlighted green is the settings that I decided to use. With this setting, we retain all CpGs that are covered in at least 40 samples by at least 10 reads. That leaves us with 4 times more CpG sites than measured by Illumina EPIC array, and corresponds to approximately 10% of the epigenome. So having post-processed my methylation calls, I was ready to proceed to finding differentially methylated regions between smokers and non-smokers. Just because we analyzed 48 samples altogether, I set the significance threshold to be 10 to the power of -5. And using these settings of significance threshold, I was able to identify 14 associations that overlapped between EPIC array and twist kit. And they are highlighted in this Miami plot in blue. And the common associations included well-known epigenetic biomarkers of smoking. For instance, AHRR, F2LR3, and ALPPL2. Overall, using EPIC array, we were able to identify 44 associations, whereas we were able to find 97 associations using the twist kit. There were some method-specific associations, and if we look at associations specific to the twist human methylation panel, we can divide them into 2 categories. The first category is the hits that were known previously. That means that they were present in EWAS catalog, but they weren't identified by this EPIC-based analysis. And the second category are hits that are completely novel. They haven't been reported before. And in terms of known hits, they include ME3, which is a malic enzyme involved in the development of liver cancer, as well as SEMA4B, which is an inhibitor of axonal growth. If we move on to novel associations, we can see there USP42, which has been associated with the development of head and neck cancer, TSPAN5, which again is associated with the pathogenicity of liver cancer. And my favorite hit, SST, which is a growth hormone inhibiting hormone. So we observed hypermethylation at the promoter of SST, which means that we are blocking the promoter. We are blocking the inhibitor. And by blocking the inhibitor, we are overexpressing growth hormone, which again may lead to cancer, and this time this has been associated with pancreatic cancer. So in conclusion, we definitely found some novel loci via twist-based analysis. We observed that the general trends in methylation are similar between twist methylation panel and the EPIC array. In terms of caveats, they include depth of coverage. You saw how I experimented with it to maximize my confidence and also retain as many sites as possible in my methylation data. Sample size, obviously, we had only 48 samples. And target tissue, it's an important caveat of all experiments that are based on epigenetics. So with this, we are coming to the end of my presentation. I would like to thank the MRC for funding my PhD, my supervisor, and the entire Marioni group, Twist Bioscience for providing kits for us to use, our sequencing team, and everyone from cohorts, Generation Scotland and Lothian Birth Cohort. Thank you, volunteers and scientists. Thank you.
We have some time for questions.
Hello. Hi, great talk, congratulations. I have a question on the differentially methylated genes between the smokers and the non-smokers. Did you do any evaluation in terms of the risk for lung cancer development?
No, not yet. We haven't done that.
Okay, thank you.
There are no more questions. Thanks, Ola. Thanks, everyone, for coming. Please help yourself with some lunch before you leave, and we will be waiting for you at the booth.